So it's a pleasure to be here, and I will also talk about the human gut microbiota. But I thought I would start off with just spending really 1 minute to think about what are the different animals like and what makes them different from each other, so that you have at least a little bit of context, and I wanted to link up to the poultry as well. So really the microbiota and the whole gut metabolism is shaped really mostly by the diet and the gut anatomy, and of course that also interacts with each other. So we have carnivores on one side, which mostly eat animal-based foods, and they're less dependent on the gut microbiota, whereas the herbivores, who really eat very plants that are very hard for mammals to digest, they are really dependent on the gut microbiota. And that is normally reflected in their gut anatomy as well. They have big fermentation chambers, essentially, in their intestinal system. And the microbiota help them really to digest the grasses that they mostly eat, so that they can really get the energy out of their food sources. And then we have the omnivores, such as chickens and pigs, and also humans, of course. So mostly you see a correlation here between the shape of the gut and the importance of the gut bacteria. But of course, nature is messy and there are exceptions. So a classical exception of this rule are pandas. And one of my colleagues at the route actually did a little bit of work with the pandas that are currently at Edinburgh Zoo. They're a gift or a loan from the Chinese, essentially. And the panda is obviously a very extreme herbivore. It only eats bamboo. It's very hard to digest for it itself. It doesn't have the right enzymes. But it has a ridiculously small gut and a really poor microbiota. So it's a real conundrum of the animal world, because there all the rules break apart. So this is really also a reminder: biology is messy and we can make general rules, but there are always exceptions of these rules as well. So I will mainly talk, or I will exclusively talk, about the human gut microbiota as well. Hervé has done a lot of the job for me on the introduction already, so I won't spend very much time. You are all aware we're highly colonized by microbes. I just want to make you aware I will only talk about bacteria, but we are becoming more and more aware that there are many other microbes as well. We have archaea in there, which is a different type of prokaryotic microbe. We have many viruses and phages. We, of course, have eukaryotes, and more people now start to study these as well. So we know much more about the bacterial microbiota than the other microbes. So just that you're aware, I think there's a whole new revolution coming up, that we better understand all the other types of microbes that also have functions. If you're looking at the bacteria, it's mainly two phyla, the Firmicutes and the Bacteroidetes, that are abundant. So what you see in this upper image here is really this sort of phylogenetic tree depicts all the different broad microbial groups you have on the planet, so anything that may live in the oceans, in the soils, and so on. So if you're looking into gut ecosystems, it's mainly these Firmicutes and Bacteroidetes that are very abundant. And then we have some minor groups such as the actinobacteria and proteobacteria. So it's not very diverse on this very broad scale, but if you home in onto these groups, so in the lower panel now you see maybe the microbiota that you may see in an individual. So each of these branches on this tree here reflects a different type of bacterial species. And you see that within the Firmicutes particularly, each individual has many different Firmicutes species, strains, genera, and families. So within these two groups, we have a high diversity, and all these microbes have different functions and different traits. So I won't dwell on this. We already heard the microbiota has many functions on the host, such as the barrier function against pathogens, immune interactions, and many metabolic reactions. I will mainly talk about the short chain fatty acid production today, but I want to make you aware also, as Hervé has said as well, there are many different metabolic interactions that are happening between the microbiota, the diet, and the host. So the short chain fatty acids are very important also in terms of quantity, but the microbiota also interacts with these phytochemicals here in the diet very much. Many different, hundreds of different plant secondary compounds that the microbiota is involved in transforming further. They make some of them bioavailable. They can change their bioactivity. But I won't have time today to talk about this in detail. The microbiota is fueled by the diet mainly. They can consume also molecules on the gut wall, so they can use some endogenous molecules from the human host, but diet really is the most important food and energy source for the microbes in the large intestine. And within the diet, it's mainly the carbohydrates that are the food source for the microbiota. And most of these carbohydrates that come from plants cannot be digested by the human host itself. We just simply don't have the digestive enzymes to break down these dietary carbohydrates. You maybe know them as fiber, or we can also call them non-digestible carbohydrates because we can't digest them ourselves. So they essentially go straight through the upper intestine, reach the colon, where they can then be a food source and an energy source for the microbiota. So we have different classes here. If you think of the plant material that we're consuming in the diet, we have the structural polysaccharides. So this is cell wall constituents of plant cells. And we also have some storage molecules that are non-digestible by us. A classic one is inulin, which I guess most of you will have heard about. You find it in yogurts nowadays or as a prebiotic. So these are fructooligosaccharides that, again, we don't have the enzymes to digest them ourselves, and they go through the system into the large intestine. We can take up sugars, of course, and then starch has a bit of a special role. We do have enzymes in the small intestine to digest starch, so principally it is available to us directly. We don't need the microbiota, but due to the physical chemical composition of the starch, some of it will simply not be completely digested through the upper gut and reaches the colon. So this fraction of the starch is called resistant starch, and I will come back to it in a minute. So I already touched on the phytochemicals. Of course, some of them are digestible and can be absorbed in the upper gut. Other ones will also go into the colon, and the microbiota is heavily involved in their metabolism. Protein and fat, on the other hand, mostly is digested further up. So on balance, we can say a plant-rich diet is more health-promoting through the gut microbiota because it fuels and energizes the microbes, and they can do their metabolism. So if you think about the non-digestible carbohydrates in chemical terms, we need to be aware that we have a multitude of different molecules here. And there are just a few examples here. This is by no means all the different molecules you will find in the diet. So I just want to, with this picture, get the message across that you have different monosaccharide components of these different fibers. We have different glycosidic linkages. So you may have alpha-linked ones like the resistant starch here. You may have beta-linked ones. And you need different enzymes to degrade all these different types of fibers. And not each microbe has it. So you need different microbes that then are able to break down these polysaccharide chains into monosaccharides that they can then be further metabolized and are used as a food, as an energy source. We also have linear molecules. We have branched molecules, as you can see here. Some of them have branch points. And then some of them consist also of non-monosaccharide constituents, such as the ferulic acid here that you find in wheat bran. So the picture just shows you there's a very high diversity, chemical diversity in the fiber, and you essentially need a diversity in the microbiota that then collectively will be able to break this down, because you need all these different enzymes to break them down and make them available. So that's on the chemical side of things, but we also need to consider the physical side of things. And this is something I think we have maybe ignored a bit too much, and more people are now thinking about this more and looking into this in more detail. So we can, if you think on the physical side of things, we can basically broadly categorize these carbohydrates into soluble and insoluble types of molecules or complexes of molecules. And it appears that the insoluble complexes are much harder to break down for the microbiota. Many microbes have enzymes that can get at these individual polysaccharide chains, but once you have it in a fragment of a plant cell wall or as a very particulate starch particle, it's much harder for the microbes to get access to these individual chains of polysaccharides and break them down. So a picture is emerging that we need keystone species in the microbiota that have these special traits that they can actually get access to this insoluble material. And I want to sort of go through a story on the example of resistant starch that emerged in our lab over the last maybe 10 years or so, where we discovered that this bacterium here, Ruminococcus bromii, seems to be one of these keystone species for resistant starch breakdown. Remember, resistant starch is the proportion of starch that simply hasn't been digested by the time your digester has gone through the upper intestine and reaches the colon. and there it is available for the microbial fermentation. Now, at the time where we discovered the Ruminococcus bromii story, already we knew of many bacteria in the large intestine that can break down starch. So there's a few examples here. Some of the Bacteroides species can do it. A bacterium called Qpcr rectale, which is one of the firmicutes, can do it. But it looks like they are mainly geared towards the soluble starches, not these particular globular particles that come into the system. So how we caught on to Ruminococcus bromii was really due to a human dietary intervention trial that we did. At our institute, at the Rowett Institute, we do fully controlled trials. So we ask a research question, and then we get our dietitians on board to design diets for us that humans are willing to eat, but that have been changed in a way that we can really tackle a specific question. And then these people eat our diets for several weeks, and we can take samples and analyze this. And the beauty is that as we provide all the diets, we have an exact idea of what they're taking in. We give them measured amounts. They do weigh-backs, so we can provide the energy dependent on their individual metabolic rate and so on. So it's very, very controlled studies. Hard to do, hard to find people who are willing to do this, eating your food for several weeks, but they're very gratifying because it's a very controlled experiment we can do in humans. So what we did in this trial, and I really only have time to give you a tiny snapshot of this aspect of the study, is that we wanted to see what difference does it make to the human physiology and to the gut microbiota if we change the type of carbohydrate in the diet. So we kept protein and fat the same. The overall macronutrient wasn't changed, but the type of carbohydrate that was in was changed. So over 3 weeks, these people were eating a diet that was extremely rich in resistant starch and very poor in all the other non-starch polysaccharides, so very fiber poor. And then in the second intervention phase, or for some people it was the crossover, in the first phase, they got all the starch taken out, and it was almost only plant cell wall fragments, mainly wheat bran. But remember, otherwise, the total amount of carbohydrate, fat, and protein was the same in both intervention periods. There was also weight loss at the end, but I don't have time today to talk about this. So just concentrate on these phases here. We had 14 volunteers. I think you see the two different groups from the crossover design. And what you see here is just a quantitative PCR assay where we looked at this specific group. Of course, we did many more measurements. I just show you this today. So we followed each volunteer over time. We took fecal samples roughly 2 to 3 times a week of these people over 10 weeks. So each line here is a different volunteer. And you see this bacterial group was completely skyrocketing as soon as these people crossed over onto the resistant starch diet. So once we bombarded their system with resistant starch, the Ruminococcus absolutely loved it and went massively up in quantity. And as soon as we went over to the next diet, they went back to their baseline levels. So that showed us the Ruminococcus seemed to be very important in starch breakdown in humans. But you can also see that 2 of the people down here didn't really have this bacterial group. And what was really exciting is when we then looked at how much starch the people were actually digesting. So remember, fully controlled trial, we knew exactly how much starch they consumed. We then took fecal samples and measured starch directly in the fecal samples. So we could see from what they've taken in what proportion of starch was excreted in feces. And you can see here for all the volunteers, the starch had pretty much completely gone. We measured hardly any starch in the fecal samples of most of our volunteers. But for the 2 volunteers here that had very low Ruminococcus bromii, we had massive amounts of starch excreted in feces. So we could see that the people who didn't have this bacterium could not really digest the starch. So that told us that Ruminococcus bromii probably had a very important function in humans in starch breakdown. And then one of our volunteers was happy to give us another fecal sample after we had made this observation. So we got a fecal sample of this person a few years after the study, and we just, in the lab, incubated it with starch. So we took the fecal sample, put it into a test tube, added resistant starch, and looked at how much of the starch was degraded over time. And that was pretty poor. Most people, you see, the starch is nicely degraded. But in this person, there was very little starch degradation in vitro in the fecal sample. We then added known individual bacteria that are known as starch degraders. So we had Bifidobacterium adolescentis, Qpcr rectale, and Bacteroides theta, and also our Ruminococcus bromii here, which had been blooming in all the people. And you can see that most of the starch degrader didn't really do very much. But when we added the Ruminococcus bromii to the fecal sample, we did restore the starch degradation to what you would see in a normal, healthy person. So that was further evidence that Ruminococcus bromii is special amongst the starch degraders in the large intestine. So the question is, what makes Ruminococcus bromii special over the other starch degraders? Well, of course, nowadays we have a lot of genome information, we have a lot of sequencing data. So if you look at the genomes of these bacteria, the question is, do you see something special? And the short answer is no, but I'll talk you through it. So basically, we know which types of genes are involved in starch degradation. There are many genes called glycoside hydrolases. These are enzymes that are responsible for the degradation of carbohydrates. And there's one group in specific, the family 13 here, GH13, are amylases. They're shown in blue here. So what you see here is that you see the numbers of starch of enzymes that are involved in starch breakdown in different bacteria. So these are all genome-based data. We scanned the genomes and counted all the genes that are glycoside hydrolase 13s in blue and also other families. So our Ruminococcus bromii is at the end here, and what you can see is that really, if you just look across, it doesn't look that different from the other bacteria that are known starch degraders. So from the coverage of numbers of genes involved in starch breakdown, you wouldn't really have picked that bacterium out based on its genome sequences alone. So what seems to be happening is that it's the way it arranges these enzymes which really matters. Sort of stalled for some reason. Here we go. So basically what this bacterium does is it organizes its starch breakdown enzymes on the cell surface in a sort of multi-enzyme complex. It has extra domains on these enzymes, which are the so-called doctrines and cohesins that can bind different proteins together. It has structural proteins called scaffoldings that then can make a three-dimensional complex, also link this whole apparatus to the cell surface. So they basically have little machines out there which can dock onto the starch because they also have carbohydrate binding domains in these little machines. And then basically through this three-dimensional arrangements of all these different enzymes that can then work together to break down these particulate starch matter, they are able to access the starch, which the other bacteria can't do because the other bacteria have a totally different way of organizing these enzymes. So just as a comparison here, we have the starch system of Bacteroides theta iota micron. It's a gram-negative bacterium, so the cell surface looks different, but it looks like this bacterium can get access to soluble starch chains, and then it fragments them and can take the oligosaccharides and monosaccharides in and consume them itself. But this apparatus doesn't seem to allow the access to very hard to digest globular insoluble starches as the Ruminococcus bromii can do. So when we discovered this, it was really interesting because relatives of this bacterium, Ruminococcus bromii, have a very similar system on their cell surface. This bacterium here, Ruminococcus champignonensis, is a human bacterium as well. But these systems were mainly discovered beforehand in ruminants, in cows and sheep. And this system was known as a cellulose breakdown system. So here we have very closely related bacteria that use the same structural way of organizing their enzymes, but the enzyme specificities are very different. So structurally, they use the same concept, but by putting a different function in, their function in the ecosystem is very different. So this is very nicely visible in the genome. So again, you have a genome-based analysis here. We have 4 different Ruminococcus species. One is a rumen bacterium here. So the blue ones are cellulose degraders. The red one is our starch degrader from the human gut. And then we have another relative up here. So what you see here is that we counted again, these different enzyme families. You have the starch breakdown enzymes here, you have cellulose breakdown enzymes in the middle, and then you have a few others dotted around. And if you just look at the colors representing the different bacteria, you can see our cellulose breakdown bacteria have many of the cellulose acting enzymes, whereas our starch bacterium very clearly stands out here. The bicirculans is the one in the middle. It likes different hemicelluloses. So here the genome coverage of enzymes tells you very nicely what the bacteria do, but they use a very similar biochemical approach in attacking these recalcitrant fibers. Okay, of course, this is all about the start of it all. What it's all about in the end is making short chain fatty acids. So they do this fiber degradation to get access at the oligo and monosaccharides that they then can ferment to short chain fatty acids. The fermentation gives them energy, and the short chain fatty acids essentially are a byproduct for the bacteria. So this is a very simple depiction of the overall ecosystem in the gut, the microbial community that turns the fiber into short chain fatty acids. But of course, there's a lot of interaction between the individual species. So just very briefly, I want to highlight that this is a community action. It's not individual bacteria doing this. So for example, if we're going back to starch now and look at a different starch degrader, Bifidobacterium adolescentis, this bacterium can break down the starch, and it makes formate, lactate, and acetate as fermentation products. And then there are other bacteria that can either live off the fermentation products. So for example, this bacterium here, Qpcr Data, can turn lactate and acetate into butyrate. You have other bacteria that can turn lactate into propionate. So it depends a bit on who's the dominant bacterium in your community, whether you get more butyrate or lactate from, butyrate or propionate from lactate, for example. And then you have other bacteria who are not very good at degrading the hard fibrous stuff, but they're actually really good at siphoning off some of the oligo and monosaccharides of the primary degraders. So this bacterium here, Rosburgia hominis, can't really digest starch itself, degrade starch itself, but it can then really benefit from some of the sugars that are fed by the bifidobacterium and can turn them into butyrate. So just that you get the idea, there's a lot of interaction in the system, there's a lot of cooperation in leading to the overall production of mainly acetate, butyrate, and propionate in the system. So we have already heard that the short chain fatty acids have a lot of functions on the host, so I won't go into detail here because Hervé has very nicely gone into this in much more and better detail than I can. All these points were already covered. So I will move on then and talk a little bit more about the bacterial metabolism that leads to the production of these short chain fatty acids. So I'm not really expecting you to look at this in detail. This is just to give you an idea of the biochemical pathways that are behind this. This is all bacterial fermentation of carbohydrates into the short chain fatty acids. You see we have different means of making propionate. There's the butyrate pathway here. The last step can be different in different bacteria. And then, of course, we have other bacterial groups that fit into this bigger picture. So what we and others have done over many years is that we looked at individual bacteria so that we could map onto this scheme to know who's doing what. So that we can, if we look at which bacteria are present, get an idea for this is a butyrate producer, this is a propionate producer. So this is just some examples. You see bacteria marched onto these pathways. So these are butyrate producing bacteria. The Bacteroidetes are very, very important in propionate production and so on. But I want to mainly show you this to also give you a caution here. For many bacteria, we can categorize them into butyrate or propionate producers, but be aware that a butyrate producer may also make lactate at the same time and formate. So they normally make a range of different fermentation acids, and it can depend on the conditions whether they make more butyrate or more lactate, let's say. And another thing is that there are a few bacteria who are not that neat, they can do different things. And probably this is horizontal gene transfer, that some bacteria have also got another way of fermentation through horizontal gene transfer. So I'm just highlighting two here. We have Qpcr catus that can make propionate and butyrate, and also Rosburgia inulinivorans. So I just show you a few data of Qpcr catus to make this point. And we've done this in the lab. If you're growing Qpcr catus on fructose, it makes almost exclusively butyrate. If we grow it on lactate, it switches almost exclusively to propionate. So just that you're aware, if you're having sequencing data from a study and you see this bacterium is present, it's very hard to tell what it actually does, because it depends on which carbohydrates are there or which food sources are there for it, whether there's somebody else that maybe produces a lot of lactate that it can benefit from, and then it's a propionate producer. From carbohydrates, it's a butyrate producer. And very briefly also, to just make you aware, it's not just carbohydrates. Also amino acid fermentation can lead to butyrate and propionate formation. Again, very complex pathways. This is not about you going in detail about these pathways, just to show you for butyrate formation, mainly glutamate is used by different bacteria through different biochemical routes. And then also lysine is a product for butyrate formation in the human gut. And then it gets even more complicated when we look at propionate formation, and many different amino acids can feed into the central metabolism of some bacteria and lead to propionate formation as well. So we don't think that's a major role in the human system, at least, because most of the proteins are digested in the upper gut, but I think it's important that people are aware of it's not just carbohydrates. I want to show you one slide of an older study from the Raud Institute that was done by Harry Flint and Sylvia Duncan, which looked into exactly this point. They gave people very high protein diets and tried to take out all the carbohydrate to see what the effect of that was. So these people were for 4 weeks on either a moderate carbohydrate high protein or a very low carbohydrate high protein diet. And what you can see here is the level of reduction in carbohydrate intakes. So especially the low carbohydrate diet really was there was no carbohydrate in these diets. This was all protein and fat the people ate. And this obviously mimics the so-called Atkins diet that some people really like to go on to lose weight all the time. But if you look at the change in carbohydrate in relation to the change in butyrate here in the middle, the butyrate levels in feces in these people dropped dramatically. So this was a 4-fold decrease in butyrate, which really is massive, because what we see in feces is only a fraction of what happens in the system, because a lot of the short-chain fatty acids are absorbed by the host. So if we see a massive change in feces, there's an even bigger change in the host. And this drop in butyrate was mainly associated with this one group of Roseburia species, which are butyrate producers. So these bacteria really need the carbohydrate to make butyrate. Protein-rich diet will not make up for it, even though there are some bacteria that can make butyrate from protein. Also, incidentally, the level of N-nitroso compounds, which are toxic compounds from protein metabolism, went up dramatically. So these high protein, low carbohydrate diets are really bad for human health, and particularly gut health, we believe, because now we don't have all the protective effects anymore of butyrate and the other short-chain fatty acids. Okay, so we did a lot of pure culture work, but of course it's a community, as I already said. We really need to think of the whole system and how it all works together. We've talked about the cross-feeding already of lactate, for example, breakdown products. We, of course, have direct competition between primary degraders. So in this example here, if we have, let's say, inulin coming into the system, we have different bacteria who can directly access the inulin. How do they interact? Who's winning out? How do they compete with each other? And then we have environmental effects. So if we have a lot of short-chain fatty acid production, then we lower the pH, and different bacteria have different pH tolerances. So the broad tendency is for the Firmicutes, which includes some butyrate producers, to be more pH tolerant. So if we're lowering the pH, we're probably increasing butyrate production in that way as well. And maybe important to remind you that the pH changes in the gut. So if you're just looking at the colon, at the proximal end of the colon where all the fiber enters the system, we have a lower pH because there's very active fermentation happening here, a lot of short-chain fatty acid production, so the pH tends to be mildly acidic. And then the system slowly runs out of energy because the bacteria consume the fiber now, less and less fermentation material there for the bacteria, so the short-chain fatty acid production slows down. The short-chain fatty acids are also absorbed by the host, so we're going towards neutral in the distal colon. And we see shifts in the microbiota. Also, incidentally, there is a higher incidence of disease in the distal colon, which could well be due to the poorer supply of short-chain fatty acids. So we wanted to see in more detail which different types of non-digestible carbohydrates or fibers influence the microbiota, which are the main beneficiaries in the microbiota if they consume these different types of carbohydrates. So we did an in vitro study, which we ran at 2 different pHs for that very reason, to mimic the proximal and the distal colon. And we tested, I think it's 15 different types of fibers that fall into these different classes. So we took fecal samples from several volunteers, incubated them over 24 hours, and then we looked at the change in microbiota. We looked at the short chain fatty acid production. And we used mostly qPCR, but also a bit of sequencing for this. So I first want to show you, and we repeated the experiment a year later with another 3 volunteers, just to see how reproducible the data were. So if I show you the short chain fatty acid data first, so we really looked in this fairly simple batch system how the whole microbiota, which short chain fatty acids were mainly produced. And these are the background levels here of the medium without added carbohydrates. Then you can see very clearly that some carbohydrates were very propionigenic, and particularly rhamnose here was led to massive propionate production, whereas other carbohydrates were more butyrogenic. So the fructans were generating mostly butyrate at both pHs, and then the alpha and beta glucans also at the lower pH. Again, the lower pH led overall to more butyrate production because the butyrate producers tend to have more acid tolerance than the propionate producers. So we can say that there are carbohydrates that tend overall to more favorable for butyrate-producing bacteria or for propionate bacteria. If we're then looking at our qPCR data, so here we have all the different bacterial groups that we measured, or species, by quantitative PCR. We have all our diets here at the 2 different pHs, and these are average data. So we looked across all our 6 incubations at changes that were significant across all volunteers. So you can see that there are some responses that are shared between people, and I'm just highlighting here again our Ruminococcus bromii, which responded strongly to the starches, which was fitting very nicely to our in vivo study. And we have another bacterium here, Anurostypes hages, which was very strongly reacting to the inulins, the fructans. And again, that tied in nicely to a human study that we'd done, a supplementation study before. So we definitely have shared responses between different people. On the other hand, if we're looking now on an individual basis based on our sequencing data, then you see a very different picture emerging. So what we did here is we did next generation sequencing of the 16S RNA gene, which is a phylogenetic marker. We can use it to really see who is there. For those of you who are not aware of how this works, you basically look at the sequence and you can put them into different groupings, these so-called operational taxonomic units that roughly represent species. So we can see by the sequence which bacteria are there, which species are there. And then we basically went through all these and collected all the ones that are known propionate producers or known butyrate producers. So I only show you a fraction of all the data here, and then it goes in decreasing abundance, so the most abundant bacteria here at the bottom. We have it for our individual volunteers at the different pHs, and down here are all the carbohydrates. So just take in the picture, don't try to read the detail, but I think what you can immediately see is that the different people look very, very different. There are some bacteria we found present in everybody, and also responses were shared on different carbohydrates, but overall, it's really different propionate producers that are present, or butyrate producers that are present in different people. So if we then basically total all these up, so if we take the sum of all the propionate producers in terms of abundance or butyrate producers, and just do a simple correlation. So now this is the sum of all those propionate producers and the propionate produced in our incubations. You see a very nice, strong correlation. And we see the same for butyrate. So all the butyrate producers together lead to a nice correlation with the butyrate we found. So basically, we have functional redundancy in the system. It may be butyrate producer A in one person that leads to the butyrate and B in another person. Interestingly, for the butyrate, we found 2 correlations, as you can see. So for propionate, there was no pH effect at all, but the pH made a significant difference in the butyrate production. So we have higher butyrate production at the lower pH, which that's now within the butyrate producers, so we think it reflects that bacteria in relative terms make more butyrate than lactate or formate or acetate at a lower pH. So that's probably a within bacterium change in their physiology at the lower pH. So we have functional redundancy. Does that mean then it doesn't really matter which microbiota somebody has? Well, I don't think that's strictly true. And you see there's still a lot of difference in different people if you look at the overall cloud in the data. And I very briefly just want to show you some data from one of our old studies on Inulin, which was just a supplementation study where we looked specifically at butyrate-producing bacteria. So what we did here is we gave people either the prebiotic Inulin or we had a control phase. It again was a crossover study. And we just took an individual fecal sample at the start and at the end of each intervention period. And then we used a marker against one of the butyrate genes. So this is just, again, a PCR-based assay against this final gene in butyrate production. And we made clone libraries and looked at the different bacteria that were present. And they were all butyrate producers because now we're using a butyrate-specific gene. So here you see the different butyrate-producing group we found in our study. And here are all our samples, which are just really organized in Euclidean distance, which means the more similar samples are clustered together. And you can see that most of the samples fell into this broad, I call it the normal range in this study, okay? The majority of people looked like this. They had mostly dominated either by Urobacterium rectale or Roseburia faecis, and then the other ones were present as well. But you can see that 3 of our people here were very different. Now, what was interesting is the people who were here didn't show a strong response to Inulin. So if you look at the volunteer F and B, all the samples are sitting up there. So it didn't matter whether it was baseline, control, or Inulin period. In the butyrate-producing community, there wasn't a big change in community. But these 3 people who had very different profiles, very unusual profiles, they showed a massive shift into that normal range after the Inulin intervention. So I think that was very interesting. Also, of course, it was a crossover study. For some people, we don't know what happened next, but this person here went from this point up here after Inulin, and they fell right back down there in the control period. They had Inulin first and then control. So they really reverted back to what they were before. It's a very small study. It's just an observation. It doesn't hold up statistically, but I think it's still very striking to see. And also for this person here, they had the Inulin phase second, so we don't know what would have happened if we had another washout sample. But when we looked at the fecal butyrate levels, this person had a dramatic increase in butyrate after the Inulin, which probably very likely was due to this shift in the butyrate-producing community. So I just want to make you aware that while many people react in the same way, we do still need to be aware of inter-individual variation. And for some people, interventions may be very important, whereas for other people, taking a prebiotic yogurt may make a little difference on a daily basis. Okay, I'm almost at the end. I just want to finish off with thinking a little bit about other factors that influence this whole thing. So we're mapping on from single strains, from in vitro work, and also from our human studies, what the different bacteria do, how the community works together in degrading different fibers and producing these different short-chain fatty acids. But, and we also then turn this into, we work also on a mathematical model. So to help us in silico to play around with the system, to understand the system better, and to design studies. So here we're basically transferring all our knowledge into coding, a mathematical coding that we can then run experiments in the computer. Just show you a little bit of data here. We had a fermentation study where we looked at short-chain fatty acid production at different pHs. Just if you concentrate on the red line, that's the butyrate. So we had a low pH phase, high butyrate production, and then a high pH phase with low butyrate production. The data are shown with the individual symbols, and the lines are the models. So if we're simulating this in the model, then we can see that we're replicating the short-chain fatty acid production very well. It's still a work in progress. We're improving the model all the time, but it's a great learning tool, and it's a great way to run experiments in silico and then take the best ones into the lab and confirm them in the lab. So I want to use the last couple of minutes or so just to think a little bit about other factors. So we can look at which bacteria do what, but we can already see from our own experience, a bacteria may do something very well in pure culture, but in the mixed system, it just doesn't do it. So what other factors are playing into how well bacteria compete in the microbiota? And I just want to highlight this point that this group of Roseburia species here, which are Lachnospiraceae, all pretty closely related. And of course, carbohydrate degradation is one thing. Which enzymes do these bacteria have? Which food sources are directly available to them? This is more data of this Inulin study I just showed you, the butyrate producers on. So carbohydrate utilization, what can they principally eat is of course an important point. Now the bacteria that are really good at inulin degradation in pure culture are Eubacterium rectale and Roseburia inulinivorans. You can see it in the name. It's the best inulin producer on its own when there's no competition. In humans, we don't see it coming up. So in this study, despite the fact that it was really high in some people, it was the Eubacterium rectale in this person, for example, that totally took over. So it just doesn't compete. Why is that the case? Well, it must be due to other traits. And one thing it could be due to in this case, for example, is the pH tolerance again. So here you see the pH tolerance of Eubacterium rectale and Roseburia inulinivorans. So this is decreasing pH, and it's the growth rate. How well can these bacteria grow at different pHs? You see the Eubacterium rectale holds up really well at the lower pH. It can still grow very well, whereas the Roseburia inulinivorans is crushing massively. So if you're taking in a lot of inulin, the pH drops massively in the proximal colon, while Roseburia inulinivorans is principally able to use inulin, it just may not be able to compete against this much more fast-growing Eubacterium rectale at the low pH. And another thing to be aware of are other growth requirements, vitamins, amino acids, minerals. Do they have any oxytrophy? So we've recently done some work with Danone where we looked at the vitamin requirements of these bacteria. So principally how we did the experiments is we basically passaged them several times through a media that didn't have any vitamins, and then we looked at how well they could grow. So with these two Roseburias here, that while Roseburia intestinalis was oxytrophic for 3 vitamins, in the absence of these 3 vitamins it could not grow. The Roseburia ficus, a very close relative bacterium in the same genus, was much less oxytroph, so it was perfectly happy in the absence of thiamine and also grew better in the absence of biotin. So I just want to conclude then, and hope I have convinced you that it really is the complexity in the non-digestible carbohydrates that are very important. We need keystone species to degrade particularly the insoluble dietary carbohydrates. And there is a picture emerging where we believe probably a more diverse range of microbial substrates, so a nice diverse fiber component. Eat all your different fruits and vegetables, try to sample from many different plant food sources, leads to a more diverse microbiota, which probably leads to a more healthy metabolite production. But I put a question mark there because, of course, there's more research needed to really confirm this. And then also I want to say again about the complexity of the microbiota. We have such a tendency nowadays to do sequencing, and that doesn't resolve often to species level, sometimes not even to genus level. If I see a paper where somebody says the Lachnospiraceae did this or that, I don't know what to make of it because there are so many different bacteria in that group that do such different things. So we need to be aware of this. Very closely related bacteria can do very different things in the system. And then also the cooperation competitiveness really depends on many factors, not just what can they do. And we really, I think, need a combination of the culture and the molecular techniques. The molecular techniques are great, but I think we need to also have these actual living units in the lab that we can work with to really fully understand the system. So in humans at least, I think inter-individual variation matters. For many people, probably responses are quite similar, even though there may be different bacteria because there is functional redundancy in the system. But also there will be people who have a very different microbiota who will respond very differently to treatment. So probably there is a call for personalized nutrition, at least for a subpopulation. And then it is a complex system. I didn't have enough time to properly talk about the models, but I think modeling, both in silico, but also with working more and more with synthetic communities to work more mechanistically on what happens is a very important thing to do. So I just want to thank all the people involved, especially Harry Flint, who has led many of these studies that I talked about today, but also all the people who did the hard work, our collaborators on the mathematical modeling side, and also our collaborators elsewhere, and of course all our funders. Thank you very much.